everyone. Welcome back to BIEB 152. This is Evolution of Infectious Disease. This is lecture number 7. Last lecture we learned about the rise of antibiotic resistance and how that is a major crisis that's coming in the future. It might be at the scale of the pandemic that we're experiencing right now. And so we need to develop strategies in order to combat the rise of antibiotic resistance. This is occurring much, much slower than the pandemic, giving us time to really think through the ways that we can combat the rise of antibiotic resistance. So I'm going to go over 6 different strategies today, but those strategies are not, it's a semi-exhaustive list of different kinds of strategies and different approaches, but lots of people are coming up with other ways as well. And as always, let's get into checking the temperature of COVID-19. And there's kind of 2 things that I want to talk about today. One is that we have certainly, and I've said this before, but I guess I need more confirmation and more data to really believe it. We have certainly flattened the curve as a country, and we have flattened the curve as Californians. This is data that was posted on the New York Times yesterday. And what these 2 graphs are showing in the top graph is, and this is for the United States, the whole country, integrated across the whole country. In the top graph, we're seeing that, over a week ago, we began to flatten the curve of the rate of new cases. So we're stabilizing at about 30,000 new cases of COVID-19 per day. In the first graph, we've seen that over a week ago, we began to flatten the curve, and that's been maintained, and that we have about 30,000 new cases per day in the United States. And so this is all good news. I do have to note that it's not, we have flattened the curve, but we're not really decreasing the number of new cases per day. So the prevalence of the disease is being maintained at a fairly steady state, it seems, in the United States, which means that more and more people are getting infected. Certainly other people are recovering from the infection, and that gives you kind of a steady state dynamic. So this is ideally the measures that we would take would actually cause the decline in the increase of cases through time. And so hopefully it does do that. As is, if we will have to just maintain this level of social distancing if this trend maintains. So if we don't actually reverse the pattern, but we actually just sort of stabilize and plateau, then we'll just have to keep doing what we're doing until we get some kind of therapeutic. So next lecture will be about therapeutics, and we'll talk a little bit more about them there. And so there is hope for therapeutics. I've also put another curve here, and this is the number of deaths per day caused by COVID-19. And what we see there is that pattern has plateaued as well. The number of deaths usually is lags behind the number of new cases, just because it takes a while for cases to become so bad that they end up causing fatalities. And so this is good news. It's nice to see that curve is flattening as well. It's sort of, the bottom graph is actually thought to be better data because we take better records of whether or not people died from COVID-19, whereas the top graph is based on, people going into the hospitals or going into their doctors and having access to COVID-19 tests, and those tests being successful. And so that is a little bit more happenstance with who actually gets tested. And so that data is not as reliable as the bottom data, but now that we see that the bottom data is also plateauing, we can be more confident that this is real, that our social distancing is working, and that we should continue it until we see that really begin to decline the number of new cases per day. So next time, like I said, we'll talk about different drugs that are being developed for COVID-19, and then we'll also talk about how resistance might evolve to these different drugs. So kind of taking what we've learned from past studies and from the lecture today, the material in the lecture today, to anticipate whether or not COVID-19 or the SARS-CoV-2 will remain sensitive to these therapies, and whether or not we have to think through how to avoid resistance evolving in the coronavirus strain. So I'm talking about this today because it was leaked last week that this remdesivir might be a promising drug for COVID-19. So this is a drug, we'll go into its mechanism of action next time, but it was a drug that was first developed for hepatitis C, but it turned out not to work that well for patients that have hepatitis C. And then it was used to treat people that had Ebola, and it had some therapeutic effects. And now it's being evaluated for its effects on COVID-19. The study was leaked last week. It was not supposed to be released yet, but we found out that at the University of Chicago, there are 113 critical patients that were treated with this drug, and most of them had actually been released from the hospital already, and 2 of them did die, but these were really severe cases, and so the death rate of very severe cases is typically much, much higher than just 2 out of 113. So this sounds promising, but I have to give the caveat that this is not a controlled study. They just had a population of patients that they gave this drug to, and they just sort of crossed their fingers and hoped that it would help them. But ideally, a trial would be done where you have a set of control patients that are very similar to the population that you're giving the drug to, and you can compare the length of time that they stayed in the hospital, the rate of mortality, and things like that between these 2 different groups. And then that's really the definitive evidence that this drug is having an effect. But this is really promising, and they have a much larger study underway in many more cities than just the University of Chicago. And so hopefully we get positive results from that soon, and the FDA clears it very rapidly for use. So fingers crossed, hopefully this all works out. I should say that this is actually, it was leaked, and that makes it feel like a tabloid, but actually this is a very reputable online magazine, Stat. So we can sort of trust this. Okay, let's actually get into the lecture today. So combating antibiotic resistance, this is our next major crisis that we're gonna have to deal with. Oh, I mean, fingers crossed that there's nothing else between this pandemic and combating antibiotic resistance. But I don't know, let's not make predictions nowadays. So, okay, strategies to combat antibiotic resistance. So I'm gonna go over 6 different strategies. The way that I'm going to build up the strategies and the way that they're organized is I'm going to start out with the simplest strategy and build up in complexity. And you also see that the simplest strategy is important to implement because it'll help stop the spread of antibiotic resistance. But really the last strategy is the only strategy where we can reverse antibiotic resistance. And so they're all important to do together in all different ways that we can combat this. But we are going to have to employ pretty sophisticated strategies. You've seen this graph, I think, twice now. So this is the 3rd time. This is just showing you that this class of enzymes called beta-lactamases is increasing through the years since we've been using antibiotics in a widespread manner around the world. And beta-lactamase, just to remind you, is an enzyme that the bacteria has that helps it resist antibiotics. So the enzyme interferes with the antibiotics, and so the bacteria then has antibiotic resistance. The reason why this is increasing through time and why the increase is exponential is because there's just such huge selective benefit for this, for a bacteria to have beta-lactamase. So think of two sets of bacteria. One set of bacteria is the evolved bacteria that has the beta-lactamase. One set of bacteria is the ancestral bacteria that doesn't have the beta-lactamase. And so now they have different evolutionary or different Darwinian fitnesses, just like we talked about in our natural selection lecture. And the one that has, the evolved one that has the beta-lactamase, is now increasing in frequency through time. And that is translating on this graph as an increase in the number of beta-lactamase enzymes that we've been able to detect in microbial populations. But that exponential growth curve should look really familiar. It's just like that R that we talked about in the selection or natural selection lecture. So strategy number one is very simple. It's very straightforward, and it's something that we absolutely have to do. It's not going to solve the problem, but it's going to make the problem a lot less bad. So we should reduce the amount of antibiotics that we use. We shouldn't use them as much in livestock production. We shouldn't use them in soaps. Soap is antibacterial itself. We don't need to add compounds to it. We need to basically preserve the usefulness of antibiotics, and the best way to slow down them becoming not useful is by just stopping using them as often. And if you have a viral infection, don't request antibiotics, don't take antibiotics. Only take antibiotics if a doctor has prescribed them for you, and you know it's a bacterial infection that is causing your illness. And so what I'm showing here is I just fit an exponential curve to the previous data, and then I estimated the R for that exponential curve, and then I just reduced that R by 25%. And so that would correspond to a reduce in the fitness benefit of these genes by just 25%. So that would translate to us just reducing our usage of antibiotics by 25%. But you can see over the years, because of the way exponential increase works, you can see that there's a major difference that accumulates. So the trend is still bad in the 25% reduction scenario, where we're still increasing the number of antibiotic resistant genes that are present. But you can see that increase is much lower, much slower. And we're not going to hit a sort of crisis moment for a very long time. So this is essentially the same concept of flattening the curve for COVID-19. We can flatten the curve with antibiotic resistance by not using them as often. And so really think through that. If you are at the store and there's two types of hand soap, one with antibiotics and one without, use the one without. Honestly, use that one. You do not need antibiotics in that product. So the question is, strategy number one, it will certainly slow the spread, as I just showed, but could it actually even reverse the problem? And so this was a hypothesis that people had for a very long time, that perhaps if we just stopped using antibiotics as often, the antibiotic resistance genes would go away. And the idea there is that antibiotic resistance genes are often costly. It costs the cell to produce the enzymes like beta-lactamase. It costs cells to produce things like efflux pumps that pump out antibiotics that we talked about. Those are very costly. It costs ATP to actually pump out the antibiotics into the environment, so to move them from the cell outside. And there's other ways that these antibiotic resistance mutations are costly. So if you mutate an enzyme that's already well adapted to perform a function in the cell, well, then that enzyme has a chance of not working as well. And so that will cost the cell, and that it won't be able to grow as fast if its enzymes aren't working properly. And so this cost, it's some jargon, is that this is pleiotropy. The way that you can think about pleiotropy is this is a side effect where there is a mutation for antibiotic resistance, but it has the side effect that it costs the cell and makes the cell grow slower. And so that's a fitness disadvantage. But that disadvantage is overwhelmed if that cell is in an environment that has lots and lots of antibiotics. So always think of pleiotropy as one mutation with two effects. The effect that's being selected for and that is adaptive, that's antibiotic resistance, and the effect that is a sort of side effect, that this cost of having that adaptation. And so the idea is that if we remove the selective advantage to having these antibiotic resistance genes, then those genes will be selected against because that cell will be paying a cost for no reason, and it'll drop out of the population. And so when we actually do a literature search for whether or not there are these costs, this pleiotropy for antibiotic resistance, we find again and again that when bacteria evolve antibiotic resistance, that we do see that that resistance is costly. And so this is just a massive table of lots of different citations. You don't have to memorize this table. It's just to show you that there's a bunch of different bacterial species that were studied. And actually, this is a fungal species, but I'm not sure. But whatever. So these are all pathogens. They can evolve resistance to these antibiotics. And here are the costs, whether or not they have a cost. Sometimes the cost is variable. So maybe it happens with some mutations are costly and other mutations are not costly. But the point is that all of these different categories yield costs, except for there are two instances where there's no cost. So just backing off antibiotic use of these antibiotics for these strains of bacteria, yeah, that wouldn't have any effect at all. But the rest of them, you would predict that, well, if it's costly, if you stopped using the antibiotics, then the cost would cause the bacteria to lose that gene and lose the function of that gene. However, that table is not taking into account that once antibiotic resistance evolves, then, and the cell is paying this high cost to be resistant, then there's a lot of selective pressure for that cell to then evolve mutations that mitigate that cost. These mutations are called compensatory mutations. So in getting a resistant gene or getting a resistant mutation, you've changed the cell in a way, and now it's paying a cost. But there's lots of ways that cells can mutate and change and accommodate those costs, actually mitigate those costs to deal with this sort of side effect, this pleiotropy. And so those mutations that deal with that are called compensatory mutations. And the effect of compensatory mutations is basically if they get rid of that cost, and you remove antibiotics from the environment, then the cell is just going to maintain that resistance gene. There's not going to be a benefit to having that resistance gene, but there's not going to be a cost either. And so that gene will maybe fluctuate neutrally in the population, but it's going to maintain in the population for a very, very long time. Microbial populations tend to be very large. So let's sort of think about what compensatory mutations are a little bit more specifically using this bar graph format that we used in previous lectures to talk about epistasis. So compensatory mutations are another example of epistasis. And so I just want to remind you that epistasis, these are genetic interactions. It's that the effect of a particular mutation is dependent on other mutations, or it's dependent on the genome that it occurs in. So in the case of compensatory mutations, it's only beneficial if it's in a genome that has that first resistance mutation. So let's just walk through what this looks like quantitatively. So say we have fitness on the y-axis and we have a bunch of different genotypes of bacteria on the x-axis. We have wild type, which has some level of fitness. When we get a resistant mutation that comes with a fitness cost. And so this is fitness in the absence of the antibiotic. Fitness in the presence of the antibiotic, it would be way up here. So this is in the absence of the antibiotic. We see that the fitness has declined, but then there's evolutionary pressure for the bacteria to compensate for that, that fitness to decrease. And so this is when that genome now has the resistant mutation and the compensatory mutation. And so the key to diagnosing whether or not this is a compensatory mutation or just a normal adaptive mutation is that if you were to engineer that bacterial genome and you removed that resistant mutation, the fitness here would just be at the level of wild type. If it was just a normal adaptive mutation, you would see a fitness increase, right? Because that mutation increased the fitness up to here, and so it should have the same effect and increase fitness up to there. But if that effect is contingent on occurring with the resistance, if it's compensating specifically for that defect that the resistance mutation causes, then its benefit is only going to occur in the genome that has the resistant mutation. So here's the scenario of compensatory mutation. This is what we would expect if it was a normal adaptive mutation, where it increases fitness to give you this, and then if you removed the resistant mutation that has the cost, then the same amount of fitness gain should occur here. That means that the benefit of the compensatory mutation was just universal. It wasn't contingent on resistance. So use these plots and the previous ones that I've gone over on fitness landscapes to really internalize what is epistasis, how does it work, what does it mean that it's a mutation effect that depends on other mutations in the genome, and make sure that you understand that. Okay, so the question is, when we have cases where resistance is costly, do those cases then result in compensatory mutations? And when we remove the antibiotic from the experimental condition, do we see that antibiotic resistance is maintained for a set period of time? And so this is the same study before that was the meta-analysis, where we have this massive table. Again, we have different bacteria and we have different antibiotic resistance genes. Then this is sort of the important column to look at. We see in most cases they maintained the resistance gene, even though selection for resistance was relaxed. There are 3 cases here. I guess you could say 2.5 cases. In this case, some were maintained and some were lost here and lost there. And so the point is that in most of the cases, you actually get the maintenance of the antibiotic resistance for a long period of time after you remove the antibiotics, in part due to these compensatory mutations. So the conclusion is that stopping to use antibiotics, or not stopping entirely, obviously you need them in the clinical setting, but not using them at home, buying livestock that we haven't treated with antibiotics. By doing all of those steps, we'll actually slow the spread of antibiotic resistance. But these genes that are already present, these mutations that are already present, will be maintained in microbial populations and remain to be a problem. So we need a little bit more advanced strategies in order to really stop the evolution of antibiotic resistance and then actually cause it to reverse. So the next strategy that we're going to talk about, number 2, is pretty straightforward, and we've already talked about it actually. We've talked about the math behind why it works. But it's that we can use drug combinations. We can use multiple different antibiotics against a single bacterial infection. And the idea there is that while the bacteria can evolve to deal with one antibiotic, it won't be able to evolve to deal with multiple antibiotics. This is an example for HIV. So combinatorial treatments are really effective for HIV. They're also used sometimes for bacteria and with antibiotics. But this is just from your section problem set where we had a mutation rate for resistance to these 3 different drugs, 3 different mutation rates. And if we were to give all 3 of the drugs simultaneously, the mutation rate would be extremely, extremely low and unlikely for the virus to evolve resistance. The same is exactly true for antibiotics. If you have multiple antibiotics, it's very difficult to get multiple different mutations to confer resistance. And so this will stop the infection. And if you wipe out the infection, then you're going to stop the spread of antibiotic resistance as well. So there is a caveat, though. It's not that simple. And so number one is that some antibiotic combinations are actually bad for patients. They can have drug interactions that really compromise the health of the patient and can become toxic. And so we can't mix a lot of combinations together. But the other thing is that they can have unintuitive interactions with themselves that interfere with their ability to work at killing the bacteria. And so for this next section, I want to talk about drug-drug interactions and their effects on killing bacteria. And so this is the first type of drug-drug interaction that I'm going to talk about. This is a synergistic interaction. And this is the ideal scenario. When you're looking at this kind of graph, this is what you want to see. So let's just walk through the graph. We're going to walk through many of these. So what we have here is on the x-axis, we have concentration of drug A, and on the y-axis, we have concentration of drug B. So this is we're indicating the MIC or bacterial growth under these different conditions. So let's just look at the x-axis for now. So when we have no antibiotics, so we have no drug A, the bacteria grow really well. So there's dark coloration here. That means that the bacteria are growing normally, they're happy. But as we increase drug A, we see that bacterial growth begins to slow down, begins to slow down, begins to slow down, begins to slow down. And then at 1, that's the minimal inhibitory concentration. So this dark line here is where the bacteria's growth completely arrests. It's the concentration where it stops growing. And so if you were to give just antibiotic A at higher concentrations than 1, we'll say this is micrograms per milliliter like before, then you would actually be able to stop the bacterial infection and your immune system would wipe it out and you would be cured. Now, let's consider a drug combination where we add in antibiotic B. So the same pattern exists for antibiotic B as antibiotic A, that once we get above this 1 micrograms per milliliter threshold, then it begins to stop the bacterial growth. So let's think about not values along these axes, but actually values where we mix both of the drugs together. So say we're to mix half the concentration of A and half the concentration of B, we would land ourselves in this cross here. And we find that, actually the bacteria growth has now been inhibited by half the concentration of each of them. So the total concentration of drug is about the same. But what we can see there is that if we move down, and now we're down to about 25% of drug A and 25% of drug B, so we have less overall drug than we would have had to use if we just used one drug or the other drug, then we see that the bacteria are still being inhibited. So bacteria are inhibited all along this line. And so if we're now just down to a quarter of the concentration of A and a quarter of the concentration of B, we actually see inhibition. And so that's why this is called a synergistic interaction, because less drug is inhibiting bacterial growth more than you would expect by just looking at the effect of A and just looking at the effect of B and adding them together, you would expect just to have inhibition right at this point or along a line between these 2 points. But now we see there's depression in this pattern, so that actually we can use less drug of A and less drug of B if they're in combination together. So people have looked at these drug-drug interactions, and they have found all kinds of different behaviors. And so synergy is what we're looking for. It's this line here. This relates to this graph here. Additivity is not bad. Additivity just means that you're not getting an extra benefit for using the 2 drugs together. They mix together fine. They don't interfere with each other. They have the exact same effect independently of one another. And so that's a fine drug combination to use as well. And so this corresponds to this graph here. But here is when the drug combinations start being bad and that you would not want to use them. And so for this drug, this combination of drugs here, this is antagonism, where now, this is the point here where you would expect, 50% of one drug and 50% of another drug would interfere with growth. But now, if you move out here and you have 75% of one and 75% of the other, you're actually still in the region. I should use this graph over here. You're still in the region where the bacteria are able to grow. Maybe their growth is inhibited a slight little bit, but they're not stopped. This is not the point of its inhibitory concentration. So certainly, if you're putting in, 1 microgram per milliliter of drug A and 1 microgram per milliliter of drug B, you would have an inhibitory concentration. But why would you give 2 drugs at the same time when that you would still see inhibition if you just use 1 microgram per milliliter of drug A or 1 microgram per milliliter of drug B? So this is an antagonism, and we do not want this kind of interaction if we're using drug combinations. Then the story gets even crazier in that there are some combinations of drugs that actually interfere with each other, so that drug A in this scenario is actually somehow conferring resistance to drug B. So this is mind-blowing. This is completely unexpected to see. But if we go over to this graph over here, I can explain what I just said. And so we have drug A at all these concentrations. And so say we have just drug B, and we're not applying any drug A into the patient or into the test tube or whatever, then 1 microgram per milliliter is going to inhibit the growth. But if you start adding in a little bit of drug A, and you add in about 0.5 micrograms per milliliter of drug A, you're now in a region where that level of drug B should have suppressed the growth of the bacteria, but now the bacteria is actually able to grow. This means that A is somehow conferring resistance to B. Maybe A is directly interfering with B, and so now the bacteria can grow, or there's some more complex thing in the cell where A triggers the cell to change its gene regulation in a way that it's no longer sensitive to drug B. So you can play through a bunch of scenarios, but the fact is that some drugs don't just have antagonism with each other, but one drug can suppress the effectiveness of other drugs. So you have to be very careful in the types of drugs that you pick if you're doing this combinatorial treatment. So I just wanted these are summary graphs, so it spells out these drug-drug interactions very specifically. And so if under this sort of additive model, this is the model where there's no drug interactions, A and B will equal A plus B. So that means that, so say I have 75% A and 25% B, I expect a certain effect of A alone and a certain effect of B alone. And if I combine them together, I'm going to be on this line. And that means that there's no no curvature, there's no kind of interaction happening. However, under the next scenario, where we have synergy, you use this line as a baseline for that's what the additive effect would be. And synergy is any deviation of that MIC line below that additive line. So this is one example of synergy, but you could have really, really great synergy down here, or just a little subtle synergy, but anything that's off this additive line and more towards towards zero is a case of synergy. Antagonism is anything that's in this direction, so the opposite of synergy. And then suppression is actually where the antagonism gets so bad that it actually is recovering bacterial growth of concentrations above which the drug alone should be able to suppress the growth. So in choosing these drugs, you have to be very careful. You have to choose synergistic combinations, and also you don't want to choose combinations, obviously, that have some kind of toxicity to the patient. So strategy number 3, so if you find yourself in a scenario where you can't just give 2 drugs at once, strategy number 3 is fluctuating antibiotics. You can't give them at the same time, but you can possibly pulse in one antibiotic or pulse in another antibiotic and go back and forth and fluctuate between the 2 antibiotics. This is true for appendicitis. People often are given Cipro and metronidazole. I should have memorized that before the lecture, sorry. They fluctuate between these 2 antibiotics, and they find that it's much better for the patient than just giving one of these strong antibiotics. These are given intravenously because if you have an appendix that bursts, you're very prone to bacterial infections that can actually threaten your life. So it's very serious, and so they do this fluctuating antibiotic regimen. Okay, so the way that these fluctuating antibiotics work is that they can actually, they sort of give a double dose of antibiotics, but they can actually even slow down the rate of antibiotic resistance evolution. And so this is a very similar graph to what we were just looking at. But now what we're talking about is an evolutionary interaction. So now we're actually talking about, the last one was just whether or not, what's the effect of 2 drugs given simultaneously on bacterial growth. And it wasn't considering antibiotic resistance evolution. Now, this is a scenario where we're fluctuating antibiotics and we are considering the evolution of antibiotic resistance. And so this is a scenario that we call collateral sensitivity. And this is really a golden scenario where if we find combinations of drugs that can confer collateral sensitivity, then we can use them in these fluctuating regimes and wipe out the bacteria and interfere with its ability to evolve resistance. Okay, so let's just sort of walk through how these drugs work together. And so this is actually a protocol in which a drug combination that has an antagonism can be used in a smart way so that you avoid reducing the effectiveness due to the antagonism, and actually even mitigate its ability, mitigate the evolution of antibiotic resistance. Okay, so let's just walk through the figure, and hopefully this makes a little bit more sense. So now the way that this figure is drawn is that this shaded region is where the bacteria are able to grow. The white region are where the bacteria are unable to grow. We have this pattern here. So this is the pattern of antagonism. So we wouldn't want to use these 2 antibiotics together at the same time, but we would want to use these antibiotics if we fluctuated from one to the other antibiotic. So it's still using them in combination, just in a smart way. Okay, so why do I say that? I say that because we now have extra information on this plot. This plot shows us the pattern of growth, the MIC and drug combinations for wild type. So this is the ancestral bacteria before it's resistant to the antibiotic. And then when it gains resistance to antibiotic A, so that's what this mutation is doing here. It's pulling out the concentration in which the bacteria can grow on antibiotic A. But when it does that, it has the side effect that now it's more sensitive to antibiotic B. And so what you can do is hit it first with A and then hit it with B, and it's going to be really, really sensitive to B. And you can fluctuate back and forth between these things so that you're actually going to be selecting against resistance mutations by applying the second antibiotic. So let's think through sort of, there is some data to support that this is true, that some antibiotic resistance mutations make bacteria more sensitive to a second antibiotic, making it sensitive to that sensitive antibiotic. And so this 1-2 punch will both wipe out the bacteria and stop the bacteria from evolving any kind of resistance to these antibiotics. And so there's a lot of people that have done these types of studies. I'm going to just talk about one study here that's really clear cut. And so the way that the study is going to be set up is much like that Lori and Delbrook study, where we're plating bacteria onto agar that has selective agents on it. So in the Lori and Delbrook, it was the phage, but in this study, we are selecting with antibiotics. That's the agent that is killing off the bacteria. Okay, so what I have here is this is just the control. This is no non-selective agar. There's no antibiotics in this agar. And so of course the bacteria grow on these Petri dishes. And if you take this bacteria and you put it into an MIC test for trimethoprim or neomycin or Cipro, you see that it's sensitive to all of these antibiotics. I've just arbitrarily shown that they're the same level of sensitivity to all of these antibiotics. So the next step of the experiment is to now plate a large population of bacteria. They've mutated, some of them have mutated, and so there's some colonies that form on this Petri dish. You can pick the colony and you can put it and grow it up into trimethoprim, and we see now, compared to before, it can grow at much higher concentrations of this drug than before. And so now you want to take that same colony and you want to put it into these other antibiotics. So remember, the colony has not seen, or this bacteria has not seen these antibiotics. It's only evolved resistance to trimethoprim. And so collateral sensitivity is where you would see this decline in the concentration of the antibiotic that the bacteria can grow in. So it's actually making them even more sensitive to these antibiotics. So there's a huge multiple step decline in neomycin, a single step decline in Cipro. But the point is that when we select for TMP, we see this side effect, this pleiotropy of becoming more sensitive to neomycin and more sensitive to Cipro. So the key here is to compare what was before they evolved resistance and what's the response after they have resistance, and we see that they become more sensitive. So here is the actual figure from that experiment. What the x-axis is number of independent colonies that they picked that had a certain behavior. And what the y-axis is change in log2 MIC. And so a 0 would mean that there's no change in the bacteria's resistance or sensitivity to an antibiotic. Above means that it's increased its resistance. Below 0 means that it's decreased its resistance. And so most of the colonies picked off of this plate. show that they have increased resistance to TMP. That's great. You know, when we select for them to have TMP resistance, they evolve it. So not great in terms of you know, dealing with the crisis of antibiotics, but great in the sense that the experiment's working. I'm not sure what's happening with this one errant bar here. Maybe this is just some kind of experimental error. And then when we take those same bacteria and we put them in two different conditions, neomycin or Cipro, we see that in general, we have a decline in neomycin. In general, we have a decline in resistance to Cipro. A decline in resistance is obviously sensitivity, increased sensitivity to those two drugs. So in fact, you know, we can have these examples of drug combinations where when they're applied in sequence, you will sensitize the bacteria to the alternative drug, and then when you hit it with that alternative drug, it'll have a hard time dealing with it. And so this is sort of trapping them with this collateral sensitivity. So this is just a summary graph of that, but now it also shows another circumstance. And so just like we had to be careful about picking the right combination of drug A and drug B when we're applying them together at the same time, we also have to pick the right combination that show collateral sensitivity and not cross-resistance if we're fluctuating those drugs and trying to avoid the evolution of antibiotic resistance. And so what cross-resistance is, it's basically the definition is in the name. So when you get resistance to drug A, you can have a different kind of response than before, where that confers resistance to drug B as well. So you can imagine that there's some antibiotic resistance mutations that are very generic and can give you resistance to multiple different antibiotics. And in that case, if you were to fluctuate them, you would, with antibiotic A, you would push to evolve resistance, and with antibiotic B, you would still begin, you would continue to favor that resistant genotype. Go back to A, now you're favoring that resistance genotype even more, and so you're just going to accelerate the evolution of that resistance. You're not actually having a fluctuating environment. Yes, the drug is fluctuating, but the selective pressure is always pushing in the same direction, driving the same high levels of antibiotic resistance. The key is to be able to fluctuate where you're pushing the bacteria one way, and then the other way, and then one way, and then the other way. That'll overall slow down the evolution of resistance. And so you have to be very, you have to have information about whether or not drugs confer cross-resistance. So one of the types of resistance mutations that often confers cross-resistance is something like an efflux pump that's very general in that it just works by expelling toxins from inside of the cell. And so that can work on many different types of antibiotics. Or if antibiotics act on the exact same protein, then often you can have cross-resistance because a single mutation will interfere with that protein in the same way and interfere with those two drugs. So those are not strict rules, and so you really have to do the experiment to see if collateral sensitivity occurs or cross-resistance occurs before you use a combination of drugs in this fluctuating regime. And I just wanted to remind you guys that this is an example of pleiotropy. Pleiotropy is where one mutation has two effects. And so this is a side effect that results in increased sensitivity, so a bad pleiotropic effect. But this is also a pleiotropic effect in that it's the side effect, but actually it gives you cross-resistance. You can, the bacteria now has higher fitness with reference to A and with reference to B. So pleiotropy is often thought to be a cost, and that's how I'll refer to it often. However, it can also be just any kind of side effect, and even a beneficial side effect like this one for the bacteria. Okay, so this is just a summary of strategies 2 and strategies number 3. It just describes what case you're picking 2 drugs here and for what case you're picking two drugs. Here, I'm going to keep moving to keep the lecture moving, but make sure to study this slide and keep it as a reference. Okay, so with strategies 2 and 3, and seeing that you can get these interesting scenarios where you have synergism, where you have collateral sensitivity, there are lots of labs out there that are going through massive libraries of antibiotics and seeing which ones have good interactions and which ones have bad interactions. Hopefully they can improve treatment strategies within hospitals. But this is often kind of a fishing expedition where they have massive libraries of compounds, and they're seeing how these compounds interact with each other and whether or not they have synergistic effects. But I want to pose the question, can we be smarter than just going on a fishing expedition? Can we actually hunt in a way that we expect to find something like collateral sensitivity between two agents that kill off bacteria? And so that's the segue to talk about strategy number 4, which is natural phages as adjuvants to antibiotics. So this strategy was implemented by Paul Turner's lab group. Paul Turner is at Yale. This is a podcast where Paul talks about some of their research and just gives you sort of an understanding of what his lab does. But Paul came up with this really great idea for how to use phage and antibiotics together to treat Pseudomonas aeruginosa infections. And actually, it probably will work for other bacteria as well. Okay, you guys might have heard about this thing called phage therapy before. And so before antibiotics were around, phage therapy, people had discovered phage, and they thought that, and they realized that they were viruses of bacteria. And they thought, we could use these to kill off bacterial infections. And so people started to use phage for that reason, but bacteria can evolve resistance to phages even faster, typically, than to antibiotics. And it was hard to culture the phages, and they had other side effects. And so people ended up just mostly using antibiotics rather than using phages to cure bacterial infections. And so phage therapy for a long time was kind of neglected by the field of medicine to focus on antibiotics. But now that we have this problem of the increase of antibiotic resistance, and sometimes patients will have an infection that just are resistant to all the antibiotics that we can throw at them, people have started to use phage as a backup. And what I'm going to talk about right now is it's a one-step advancement on phage therapy, where you use phage in combination with antibiotics in a very smart way. And this strategy has cured many patients actually now, and I think is a really good way to go about using phage and developing therapies with them. So the idea that Paul and his lab had is that we know that efflux pumps confer resistance to lots of antibiotics. We know that pathogens like Pseudomonas aeruginosa do have these efflux pumps. The efflux pump that they focused on was this OPRM. And so here is the protein structure of it, and here is how it works within the two membranes of the bacteria, where it has a system where drugs come in, and there's chemical reactions that facilitate the drug leaving the cell. And where the drug leaves the cell through is this OPRM protein. So we know that this is critical. If you don't have this pore to get rid of the drug, then these genes here, these proteins here, won't be able to actually expel the drug from the bacteria. And what we also know is that these types of proteins on the outer membrane of bacteria tend to be targets of phage. And if the bacteria are resisting the phage, the way that they do that is they tend to get mutations in these proteins to interfere with the formation of the protein, so that basically the phage can't even sense the cell. It's unable to bind to the cell and to infect it. And so what they thought is, this creates a dilemma for bacteria. They can either get rid of OPRM with mutations that confer resistance to the phage, or they can have OPRM and have resistance to the drug, but they can't do both of them at the exact same time. You have to choose one or the other. And so this is having information up front where you predict that you should get this collateral sensitivity behavior, that if you evolve resistance to the drug, you become sensitive to the phage. If you evolve resistance to the phage, then you become sensitive to the drug. And so this was their idea. And what they wanted to do, though, is find a phage that actually used OPRM and was able to infect Pseudomonas aeruginosa. And so they weren't sure if they were going to be able to discover a phage that had this property, but they went looking anyways. And they had a very smart way to look for this phage or to screen for this phage. So what they did is actually, it's a crazy story. They went down the hallway to a colleague that had some lake water, and they knew that there's lots of phages in lakes, there's phages everywhere, there's phages in your gut, there's phages in the soil, and they just thought, okay, this is easiest to just look in lake water, we can filter it and just isolate phages, and then challenge the phage to be able to grow on Pseudomonas aeruginosa. And so that's what this plate is showing you guys here, is that this is a plate and we have all of these plaques. So these plaques are cases where a phage has landed on the plate, and this is a lawn of Pseudomonas aeruginosa, the yellow coloration. And now we have a clearing, which is the plaque. And so that represents a single genotype of phage that was able to infect the bacteria. There's many more phages that were put onto this plate. Where there are spots are just positions on the plate where a phage that landed that was able to actually mount an infection and create this plaque. And so they say, okay, all of these different spots, these are phages that are able to kill Pseudomonas aeruginosa, but we want one that is not just able to infect aeruginosa, but is able to infect it through that OPRM gene, that outer membrane protein that's used in the efflux pump. And so then what they would do is they would take these and they would take a small portion of the phage from here, and they transfer it to a plate that had Pseudomonas aeruginosa, but it had the gene deleted for this protein, so we know that there's no way that that protein is on the outer membrane. And so if the phage can infect on this plate, but can't create a plaque on this plate, that means that phage, yes, we know is able to infect aeruginosa, but is unable to infect this strain because it's lost what we call a receptor. It's lost this outer membrane protein that the phage uses to infect the cell. And so that's a phage that we want to use, where it's going to pressure the bacteria to lose OPRM. But if it loses OPRM, then the antibiotic is going to get the bacteria. So if we use a combination therapy of the phage and the antibiotic, then we know that the bacteria are going to die because they have to do one thing or the other. Both things at the same time are going to kill them, and we'll have that collateral sensitivity and that really nice therapy. And so most of the phages they looked at probably were able to create plaques here, but there was luckily this one phage OMK01, that was able to produce a plaque on this plate, but not this plate. So here is just one of the figures from their papers demonstrating that the combination of both the antibiotic and the phage put the bacteria in a terrible situation that causes it to not be able to grow and to be wiped out. So we have time on the x-axis, and we have density of bacterial cells on the y-axis. So higher density means that the bacteria are able to grow. And so this trajectory here is showing you what the bacteria do when there's just antibiotic present. So I like this figure, but it's sometimes hard to keep track of what's going on. So this box here corresponds to this color. So you have to match the color to the lines. And what it's showing you is that if you take the Pseudomonas aeruginosa and you culture in the lab and you look at the bacterial density through time, you see that even in the presence of antibiotics, it has some level of resistance because it has the efflux pump. That's what this is showing here, is that this cell has the full efflux pump, including the outer membrane protein, the pore that expels the antibiotic. And so it's resistant and it grows just fine. And then if you add in the phage to the same bacteria that has the full efflux pump, now you see that the bacteria begins to grow, and it's now facing this sort of evolutionary dilemma as the phage is able to infect it and the antibiotic is acting on it, and it just drops down to a very low level, probably the level of detection really in the machine. And so that's what you want to see. It's much, much lower density than with just the antibiotic alone. And then these bottom two are just two different controls. One is both are the cases where you don't have that outer membrane protein, and so the pump doesn't work properly. So the strain of bacteria being used is the one that is sensitive to the antibiotic. And so the antibiotic is suppressing its growth. It's resistant to the phage, and in this case, there's no phage at all. And you just see that the red line and the yellow line are the same. They're both getting killed off by the antibiotic because they don't have that functional outer membrane protein. So the green in the long term looks just like these negative controls. So that's exactly what we want to see. And it suggests that the action of the combination therapy here is that the bacteria are trying to avoid the phage, and so they're evolving resistance by getting rid of this outer membrane protein, and so then they're no longer able to get rid of the antibiotic, and so they're behaving like this square down here, which is what we see in the experiment. This certainly does work. This is just walking you through those steps that I've already gone over. So this is just for your notes. And they successfully administered it to this Yale doctor who had a Pseudomonas aeruginosa infection that was multi-drug resistant, and the doctor's health was failing, and they couldn't get rid of the bacterial infection. They administered this phage and the antibiotic, and this guy ended up recovering. There's an article about this in STAT that we talked about earlier. Definitely check it out. It's a pretty interesting case. So since this, there has been other trials where other patients have been successfully treated with this combination therapy. I should note that also at UCSD, we've had patients where we've used other phages to cure their infections and have been successful as well. So Yale is a hub for this kind of phage therapy, but certainly UCSD is as well. And actually, we have the first phage therapy institute in North America. So this is, I think this is a growing field in case you're interested in this stuff. So this is just a slide that summarizes a little bit of the history of phage therapy. I've already basically gone over this. Phage were discovered independently 2 separate times, once in 1915 and once in 1917. They were used in World War I on patients to cure them of infections. They were very helpful in World War I. And UCSD has the first North American Institute for Phage Therapy. It's called IPAT, if you want to look that up. Okay, now Paul Turner's approach relied on being lucky in finding a phage in the environment that would have this synergistic interaction with antibiotics. So Tim Liu, a while ago now, actually published a paper where he engineered bacterial phages so that they had synergistic killing effects on bacteria. And they actually even cause bacteria to have a slower rate of evolving resistance. And so this is kind of maybe even a little bit smarter in that we don't have to depend on finding that lucky phage, but we can engineer in characteristics into that phage, genes, adding new genes into the phage in order so that they alter the bacterial cell in a way that then makes the bacterial cell sensitive to antibiotics. And so you give them the sort of 1-2 punch to wipe them out. This is what we'll call a platform. It's a synthetic phage. That just means we've engineered it. We've engineered its DNA. And it's a platform in which we can deliver lots of different genes and have different synergistic effects. And so we can develop it in lots of unique ways to interact with different antibiotics or different bacteria and so forth. So I'll just talk about this one case where they added this gene called LEX-A3 to the phage that altered the gene expression in the bacterial cell in a way that had a synergistic effect with antibiotics to kill off the cells at a higher rate. And so the way that this works, and this is called bacteriophage M13, and they can take this bacteriophage and they can add in other genes to the bacteriophage that they know will alter some characteristic of the bacteria. And so when this phage infects a cell, what it does is it doesn't kill off the cell, it just mounts an infection where more of the phage can grow and propagate from the cell, but it doesn't actually lyse the cell and kill it. This is an infection much more like we tend to have, where we'll have the common cold or flu, and we typically survive that infection. But this is also what you'd call a chronic infection, that it's very hard for the bacteria to get rid of this phage once it has it. Okay, but the reason why this phage was chosen is because it puts DNA into the cell, and you can program that DNA to express these new genes. And so it's expressing this LEX-A gene, which alters gene regulation. So let's rather than starting from here, so we have this new gene in, let's start from this point of the diagram. And so what they're doing is they're assaulting the cell with these bacteriophages and also an antibiotic. The antibiotic causes DNA damage. When cells experience DNA damage, they're not helpless to just to sort of lay there and get attacked by this antibiotic. They can actually upregulate a system that turns on DNA repair mechanisms. So this system is called the SOS system, and it triggers the DNA repair system, and then that DNA repair system fixes the effect of the antibiotic. So what this graph is showing you here is that in red, this is just the effect of the antibiotic alone, where you would have cell death, some cell death. But when you have this LEX-A added to it, the effect of LEX-A is now described in blue. What it does is it actually suppresses the SOS system, so stops it. And so then what happens is it stops the DNA repair. And so now any death that's caused by the antibiotic because of how it's damaged the DNA is now enhanced. And so there's a lot more killing, and the bacteria has a hard time to overwhelming, the whole bacterial population gets overwhelmed by this combination therapy. So here are the data from this one trial. So we have mean killing effect, and so a 0 means there's a 0 flat across, so the x-axis is time. A 0 across here would just mean that there's no growth of the bacteria and there's no killing of the bacteria. So under these experimental conditions, there's just very subtle growth if you don't have the phage or you don't have this antibiotic. And so if you add in just the antibiotic, you see that the bacteria is certainly sensitive to the antibiotic, but it doesn't cause the population to completely crash out, and that's likely because there's some level of resistance that occurs in the population. And then if you add in the phage without the LEX-A gene added to it, and you also add in the antibiotic, you can see, okay, well, yeah, combination therapy with antibiotics and phage also work because the phage, it's not good for the bacteria to be infected by this phage. And so we see that there's enhanced killing if we have both the phage and the antibiotic. Okay, so that's good. But remember, we can engineer the phage in a way to have a synergistic interaction to kill off the bacteria even more. And so that's what happens when we add in the antibiotic and the engineered phage. We see multiple folds of decrease of the bacterial population, really just wiping out the bacterial population from this test tube. And so it's unable to evolve resistance, and it's overwhelmed by this synergistic interaction. So like I said, this is just a platform. You can add in different genes. You don't have to just rely on LEX-A. LEX-A just worked very well by altering the gene regulation of the bacteria. But I think that's kind of, for me, that's a little bit confusing. You know, like how exactly are we changing the gene regulation? And I guess it makes sense in light of the SOS system. But there's also other more direct ways to even resensitize bacteria to antibiotics, so that then you can hit them with the antibiotic. You can resensitize them to therapies that were originally they were resistant to and were not effective. And so one way to do that, and one way that you can imagine, one gene that you can imagine adding to this M13 phage is a gene sort of for the pore that antibiotics enter the outer membrane of the cell. So this is a diagram from an earlier lecture where we talked about different ways of evolving antibiotic resistance. And one of the ways is that you could get rid of this pore, and so that you stop the antibiotics from diffusing out from the environment into the cell. And so then they are no longer affected by the antibiotics. And so if you have bacteria that evolve resistance by getting rid of the doorway of the antibiotics into the cell, you could put that gene for the protein for the doorway into this phage, so that then the outer membrane of the bacteria is now receptive to that antibiotic, and those antibiotics come into the cell. So that's sort of one alternative strategy, one new gene you could add to this platform. And there's lots of different ways that you could add in genes. You could add in genes that destroy biofilms. So a lot of times a problem with treating infections, especially like in the lungs, if you have a bacterial infection, is that they tend to make these sticky biofilms that clog up your lungs. And then those biofilms actually provide some protectant to antibiotics, because it's hard for the antibiotics to diffuse through the biofilms. And so you could actually put in genes in the M13 phage that interfere with creation of biofilms. You could put in other genes that increase the permeability of the outer membrane to antibiotics. And you could put in other genes that affect gene regulation, maybe in ways that stop efflux pumps from being expressed. And you can just sort of imagine all kinds of different ways. So this is a generic platform that we can use to create these synergistic interactions with antibiotics. So the sixth strategy I am really impressed by, and it comes from labs at UCSD in this Tata Institute for Active Genetics. This is a new building on campus that the Institute is housed in. This is just an article about the opening of this institute. This is Pradeep, our chancellor, and this is a professor, Ethan Beer, who created this thing called a gene drive. He didn't come up with the idea of a gene drive, but he and his lab members created the first gene drive. They actually created this genetic element that I'm going to describe how it works in a second. in fruit flies. But recently, he's teamed up with Victor Nizet in his lab, and they have created a gene drive in bacteria. And the way that the drive works is to actually reverse antibiotic resistance. It is this genetic element that can track down antibiotic resistance genes and kill them off. So it's really interesting. It's really powerful. It's just at the first stages of being constructed. It's very far away from ever being used, but it really, these kinds of smart strategies give me hope for stopping the spread of antibiotic resistance genes and actually reversing the spread, getting rid of the antibiotic resistance genes. Okay, so this is a little bit molecular focused, a little bit complicated, but what a gene drive does is basically it cuts DNA that you don't want, and then it pastes in DNA that you do want. And when it pastes in DNA that you do want, that DNA has the drive encoded into it. So then that copy of the drive can then cut and paste itself into other bits of DNA. So let's sort of step back a second and think about what I'm talking about here. And so there are a couple different components in what is called a gene drive, and I'm particularly talking about the gene drive that has been constructed for bacteria. It's different for flies, of course, and other organisms, but this is the one for bacteria. And so what we have here is this Cas9 protein. And so if you know anything about biotechnology, you know that this Cas9 protein is completely changing the way that we're able to edit genomes and do all kinds of sophisticated things. Cas9 they're trying to develop strategies to use Cas9 in ways that we can more efficiently and much cheaper diagnose somebody with having COVID-19. And so this is a sort of general all-purpose enzyme that people are using in lots of different ways. But at the essence of Cas9 is it's an enzyme that cuts double-stranded DNA. So there's lots of enzymes that cut double-stranded DNA. What's unique about Cas is that you can program the enzyme to cut double-stranded DNA in a particular location. And so you use this guide RNA here. This is a little piece of RNA that has a sequence here. So this is, A, G, U, A, C, C, so forth. And that tells the enzyme it matches up using Watson-Crick pairing with a sequence in the DNA of, say, a bacteria cell, and it matches up and it cuts it in a very particular location. And so this guide RNA programs the enzyme to cut the DNA in a particular location. That location could be an antibiotic resistant gene. And that's been shown to be successful, that you can program this, cut that gene, and then you get rid of resistance in the population. But you can use just Cas9, but you can also couple it with this second system called lambda red, which then causes what we call homologous recombination. You can think of this as just pasting in. So you have a sequence of DNA that you want to paste in to this cut. So you want to remove the antibiotic resistance gene and then paste in the drive DNA, and then that cell is no longer going to be resistant, but it also has the genetic material for a drive, so that then if a new antibiotic resistance gene comes into the bacteria from horizontal transfer or some other mechanism, then that drive will cut that new invading gene. And so this is an active system that is always surveilling the bacterial population from evolving resistance to the antibiotic that you program it to assault. Let's keep going so you can understand a little bit more about how this works. And it is complicated. There's lots of information online if you really want to understand how drives work. There's really cool technology. And I see a big future with it. And especially if you're looking for research at UCSD, there's lots of people that work on gene drives. So this is a drive here. H1 to H2, all of this is DNA, and it's encoding different genes and different properties and allowing this drive to work. This is in this larger plasmid DNA, so this is just its vector to help it replicate and to move it from one cell to another cell. And so you have this Cas9 and this gRNA. So this is saying that it's encoding for that enzyme that is programmed with this piece of RNA to target a particular spot in the genome. In the case that we're going to talk about today, it's targeting an antibiotic resistance gene, cutting it in half so that that resistance gene no longer works. Then the next step is homology-directed repair or recombination. So this lambda red is turned on, and what this lambda red does is it stitches DNA together so that if you have a sequence of DNA that's identical to another sequence of DNA, even though they're on separate strands of DNA, what it does is it stitches them together. And so we have in our gene drive, we have what are called homology arms, H1 and H2. These are pieces of DNA that we've put in here that we know are identical to the antibiotic resistance gene. And so what we know is going to happen is that when this cut occurs and we upregulate lambda red, then this whole piece of DNA is going to be converted into this segment of DNA here. And what you're going to get is now you have two copies of that drive. One copy of the drive in the plasmid and one copy of the drive in the genome or another plasmid that had the antibiotic resistance gene. But now we've just cut that antibiotic resistance gene in half, replicated this gene drive, and now that gene drive can go on and spread to another antibiotic resistance gene if you have multiple copies of that antibiotic resistance gene. That's very common. Often in bacteria, you'll have multiple different plasmid pieces of DNA. So these are just really small bits of DNA that occupy the cytoplasm of bacteria, and you'll have maybe 50 copies of them. And so you need a genetic element that can spread to one, not just one copy, but can replicate itself and spread to multiple copies. And so the gene drive actually works really well in this context. So here is just a summary slide giving you more specific definitions of all of these elements just in text so you can study. And so the idea with the drive is that they've developed it so that they show that it actually works. And if you start out with a population of antibiotic-resistant bacteria, you can sensitize the bacteria to antibiotics and then hit them with the antibiotics, and then they just die off. And in the future, what we really need to do is to be able to put the drive, so this is the drive on a plasmid, put it into some kind of vector or some kind of genetic element that will allow it to transfer from one bacteria to another bacteria. And so if it has that property that can actually transfer from one bacteria to another bacteria, then it can spread through bacterial populations and wipe out the antibiotic resistance gene in that full population. And so the way that you can do this, there's multiple different strategies, but we talked about one mechanism for horizontal gene transfer is conjugation. And so if you have this drive that's coupled with conjugation, you can actually have this bacteria that has the drive in its genome. It's also in this plasmid. Then the plasmid can spread to another genome that has the antibiotic resistance gene, and then that genome can be cured of that antibiotic resistance gene, sensitizing this bacteria to the antibiotics. So this drive is more complicated, but it can actually, it's predicted to be able to spread through bacterial populations and cure them of these antibiotic resistance genes. Okay, so we have 6 different strategies we can use to combat the rise of antibiotic resistance. The first is the easiest to understand, just reduce your use of antibiotics, and so the problem won't accelerate as fast. That's flattening the curve, but for antibiotic resistance. We have drug combinations at the same time or fluctuating. We have phage collateral sensitivity. So it's number 4 is a play on number 3, except you're not using 2 different antibiotics on the bacteria. You're using antibiotics and phage on the bacteria, and you're picking the phage in a way that's smart so you get collateral sensitivity. The 5th one is using engineered phage that have these synergistic interactions with antibiotics. They're adjuvants to antibiotics. And the last one are these gene drives where we can actually put this genetic element into microbial populations and hopefully wipe out antibiotic genes from the whole population, resensitizing them to our therapies, and hopefully reversing the rise of antibiotic resistance and preserving the effectiveness of our therapies. So here's just another summary to talk about, the way that I try to organize this lecture is that we start out with the sort of simplest to the most complex, the sort of oldest idea to the newest idea. And I wanted to say sort of where are these in the development in fighting antibiotic resistance? Well, approaches 1 through 3 have been approved to use in humans. 4 has been approved only for compassionate care. 5 is still under development. It's contentious to use synthetic organisms to treat humans, and so that we have to get over that hurdle and make sure that there's no problems there. And number 6 is just at the very first stages of development, but number 6 I think has a lot of promise. Okay, thank you guys very much, and I'll see you guys on Thursday.